Hello, everybody. This is Dr. Newman. We hope that these talks are going to get on the internet out there one day. This is the beginning of the neuroscience course here at Einstein Neurology Residency. This is the first of a 3-year course. The course is designed so that by the end of the 3rd year, you will have the equivalent education of a first-year graduate student in neuroscience. The courses are basic science. They are not intended for clinical purposes. They will, of course, be used that way by you later. That's okay, but that's not what this course is about. This year is neuroanatomy. Next year is neurophysiology. We used Dale Purvis's book. And then the 3rd year, sort of a 2-semester course. The first semester is neurochemistry, mainly macromolecular, some small molecule, and then there's neuropharmacology, right? So we begin neuroanatomy. We're going to begin today with the neuron. Most of these lectures are very, very focused in their topic. There are about 30 lectures that we're going to get through. The approach that we're going to use is that of Truex and Carpenter. So if there was one reference book that you wanted, I would say try to find an old copy of Truex and Carpenter that's no longer in print. And the 9th edition was the last edition I'm aware of. The 8th, and that's not available. The 8th edition is the one that you want to look for if you want to do this. There are many, many others. But this is largely out of classic books, including, for instance, Alf Brodel's book on neuroanatomy, and supplemented with a wonderful neuroanatomy book by Joseph Martin 10 years ago. All right, let's just get to it. So here's our maybe. So, all right, here we go. Our outline is basically, I'm going to give a brief introduction to the neuron, and then we're going to look at histologic techniques. So this is going to be important, then we'll apply for the rest of the course. We're dealing with a neuroanatomy course. That means we're going to be looking at tissue. And the tissue in most cases is not living, breathing tissue inside the head. And of course, this is heavily focused, I should say, on human neuroanatomy. Rarely will we take in other anatomy, sometimes, but only to make a point. And the other thing I would say about it is that this is central nervous system. We are not going to deal with the peripheral nervous system in this course at all, really. We will then look at varieties of neurons in this lecture, and then we're going to take up the cell theory of neuron here, which includes dendrite, cell body, axon, and synapse, and we'll get to all this. The nervous system, these are some basic statements here to begin this course. The nervous system consists of neurons and glia and a supporting mesenchymal tissue. We're going to think embryologically frequently during this course. We will have separate lectures on embryology, but the concepts of embryology will be everywhere. In virtually every lecture, we will see the effects of embryology. The relationship of the neuron and the glia is inseparable. You will see that on the last slide of this lecture. It's simply inseparable, and it's not completely understood. It's a great topic and a great area. I should also make the point that neuroanatomy is a field of active research. This is not static, and we learn new things all the time, and I try to keep these talks updated. And then the last point that I want to make is that everything that we see ex vivo, that is when we're looking in the microscope or whatever technique we're using, it's an artifact. We only get to see artifacts. We don't get to see the natural tissue. We don't have access to it as it lives and breathes and pulsates in our head. Now, the concept of neuron seems so embedded in your consciousness that it's probably going to be a surprise to know that this functional neuron hypothesis was first proposed by David Brody in 1962. That's only 60 years ago. The first time I gave this talk, it was only 20 years ago. So this was like new stuff. And you go, what? But Bodian was the one who sort of synthesized all the information that had been coming since the 19th century, really. He talked about a dendritic zone, a cell body, an impulse generating site, and the axon. And that just seems so natural that you think it's like, how could it have ever been that no one thought about this? But if you look in the literature of neuroanatomy in the 50s, I'm talking about the active research, people are still talking about a reticular nervous system, an organized nervous system, which was a network, literally a network. And Bodian was the one who focused us on the notion that that may be true, but the unit of the network is definable, and that unit is the neuron. Okay? So all derived from this, and this is just going to seem so ordinary to you in this day and age, but it wasn't, is that you have a dendritic zone which receives the input. That's how the neuron's going to get its input, is through the dendritic zone, which is not entirely synonymous with dendrite, because sometimes the cell body can have synapses and also contribute to the dendritic zone. So, of course, most of it is the dendrites of the neuron, including their spines, which we'll look at today, but also the cell body, and then also specialized receptors, which we'll also look at a little bit today. All of those are the dendritic zone. The cell body in this hypothesis was the pericardia, if you want, really focuses around embryology. It's how the neuron develops and migrates to where it's migrating to, and then sends out the dendritic shoots and the axonal shoot, okay? And all of that being ascribed to the pericardia, and then the maintenance, and if necessary, the regeneration of those structures is ascribed to the cell body. So in this hypothesis, the cell body is mainly to create and maintain the extensions of the cell. The impulse generating site is in the central nervous system, is typically the axon hillock, all right? And in motor neurons also the axon hillock. There are axon generating sites in the central nervous system within dendrites, so that can happen. And in sensory receptors, and I just looked this up last night, I was like, what? In sensory receptors, the impulse generating site is at the first node of Ranvier after the receptor complex. So that was sort of interesting to me, and analogous to the generation at the axon hillock. And we'll look at the axon hillock and talk about it. And then, of course, the axon is that portion of the neuron which transmits the action potential away from the dendritic zone and cell body, okay? And then terminates in the synapses and the specialized endings. You can interrupt with questions anytime you want. That's sort of it for the introduction. Now, I want to spend a little while on histologic techniques. And I was amazed when I went over this. I have actually used every one of these techniques during my research at early stages with brain slices and with rat brain. So piece by piece, first of all, the first thing that you have to do when you, to prepare the tissue for viewing is fix it, right? If you don't fix it, it'll be mush by the time you get there and you're not going to see anything, okay? And tissue preservation then is really the purpose of fixation, its main purpose. It kills bacteria, and equally importantly, or more importantly even, it inactivates autolytic enzymes, okay? So otherwise the cell is going to engage in apoptosis and destroy itself, and you don't want that. Good fixation minimizes shrinkage or swelling or other distortions, but this is not always possible, right? But there are other points of fixation here. Fixation prepares the tissue to receive specific dyes. If you use the wrong fixatives, you don't get to create any contrast under the microscope or whatever technique you're using. So it has to permeabilize the tissue to receive specific dyes or labeling techniques. It also enables embedding, sectioning, and mounting, all of these from fixation. Here are the two. Basically, fixation falls into two categories. There's perfusion fixation and immersion fixation. If I were doing a rat study and I wanted good detail, I would take the rat and I would cannulate its aorta, I'd anesthetize it, paralyze it, usually with ketamine. You then cannulate the aorta and infuse a fixative, formalin or whatever you want to use. And in a few seconds, the whole rat simply is fixed. And then you just pull the rat, you pull the brain out of this fixed rat. The whole rat is fixed. And the fixation is spectacular then under that circumstance, because the fixation was done instantaneously upon the animal's death. All right? We don't get that. In humans, that obviously is not the case. And in humans, what you do is you get an autopsy, and the body sits for hours at room temperature, okay, or higher if you're not lucky. And then you remove the brain and you fix it in a bucket of formalin. All right, so this is an immersion fixation, and you're waiting for the formalin to penetrate the entire brain. And you can imagine that that isn't always optimal. That's what we get to look at in humans. The fixatives that we use there are multiple ones. This is just samples of typical stuff that we use. The one that you smell in the autopsy suite is formalin. That's mainly formaldehyde in water, and then it's diluted. But you can use alcohol. Sometimes you use multiple fixatives, one after the other, a sequence of fixatives. For instance, if you use alcohol, it will remove the lipids. Sometimes you want that, and sometimes you don't. It might, depending on what you want to look at. If you want to look at myelin, you don't want to remove the lipids. If you want to look at the axon, you might want to get rid of the myelin, okay? Glutaraldehyde is the typical fixative that you use for electron microscopy. And nearly all of the time when you look at transmission EM, we will have used glutaraldehyde for the fixation. Sometimes you freeze the tissue for fixation. Sometimes you want to do histologic techniques. We'll look at that in a minute, where you want to look at enzyme activity, not just the presence of the enzyme. But you actually want to look at the activity. You can't fix it in glutaraldehyde or formaldehyde. And so we use liquid nitrogen fixation. Another way of fixing the rat is to anesthetize it, crack open its skull with a pair of scissors, with forceps, pull it apart, and just dump the brain into liquid nitrogen. Okay, that's not quite as good as perfusion fixation, but it's pretty fast. And that liquid nitrogen penetrates the relatively small rat brain quickly. And so you will see, we are going to look at today, and almost every time we get together, we will look at tissue which was fixed this way. All right, and I'll point it out when we get there. All right, and typically the scanning electron microscopy or the histologic techniques are going to be fixed that way. We have to embed the tissue. You almost never simply take a piece of brain and section it. You virtually always have to embed it in a material which will hold it together, so you can lay it on your slide or on your disc for EM, or wherever you're going to put it, over the emulsion that you might be using for radio labeling. Of course, that's another important histologic technique. The vast majority of light level histology is done in a simple wax called paraffin. If you ever have seen these big blocks of tissue where you have whole brain and you're looking at the whole brain slice in this piece of clear plastic, that's collodion, all right, is your fixative in that regard, all right? For electron microscopy, you use EM resins, and then for frozen tissue, you use a material called OCT, and you just drip it onto it, and the tissue is sort of on it, and then you add more and it penetrates, and now you section off with your cryostat sections. Of course, now we progress on to stains, which is sort of probably where you thought we would start, but we have to have the tissue ready for the stain. And then from the stains, the H&E is still used, although not that much, but you will see a lot of typical H&E, okay? Luxol Fast Blue is the standard light level histology stain for myelin. I'll show you that in a minute. Crescent Violet is what you use for the neuronal pericarya, because the crescent violet binds to nucleotides, DNA and RNA, all right? And then we're going to play with silver stains a lot. Bodian used a silver stain. You're familiar already with Golgi. And Ramon Cajal, who in many ways is the father of modern neuroanatomy, used a silver stain, which bears his name. For histochemistry, now histochemistry, sometimes you're looking at the metabolism of the tissue, but sometimes you want to see what enzymes were present, what receptors were present, that sort of thing. But you also might want to find some small molecules which are present. And so we have a variety of techniques, I'll show you some in a second, which can look at either enzymes or small molecules. You can also do, especially in this day and age, there's a whole world of immunohistochemical labeling, which is phenomenal, and you get to incredible levels of specificity. Then you, of course, you don't see the antibodies, so you have to, however, find a way of labeling the antibodies, either by coupling them to an enzyme or coupling them to colloidal gold or that sort of thing. Track tracing is a particular histologic technique, which is great. You begin by injecting the rat paw, would be typical, or the whisker barrel neurons in the face of the rat, and then with horseradish peroxidase or with a viral particle, and wait a period of time with the animal alive and let it be retrograde transported, and then identify its presence up in the brain. And that way you can find the neurons which are responsible for that sensory area, or spinal cord for that matter. We used to use a fair amount of radio labeling in my research, usually C14, sometimes tritium, rarely P32. I never used 23 sodium. And this is done by preparing the tissue for a light level anatomy and then laying an emulsion over it with the radioisotope in it, and let those radioisotopes activate the emulsion. The C14 is in the tissue and you catch it with the emulsion. And then for electron microscopy, the classic stain is osmium tetroxide, and we'll see that. If you're going to use in scanning EM, we use atomic gold, which is sprayed on the tissue at a low angle, and I'll show you pictures. It's just easier with the pictures in front. Okay, this is just this is not, we're going to look at this neuron in a couple minutes to look at the neuron structure, but that's not what we're doing right this minute. All I want to show you is cressyl violet with Luxol fast blue. And the cressyl violet is creating this tigroid appearance of what's known as the Nissl substance. Nissl was the first one to use this, and he noticed this tigroid appearance in the cell body, and he called it the Nissl bodies, okay? And the neuron and the nucleolus of the neuron, right? A lot of DNA and RNA staining in there. And you can see an axon and a couple of dendrites. You'll notice that there is a fixation artifact of a space around it. Oh, well, you know, oh, well, that's the way it goes. And then you have all these pale blue lines, which is myelin, because this is also stained with Luxol fast blue. So this is both cell body. This is a lovely set of pictures that you get out of this combination, where you can see the cell body and you can see the axons, the myelinated axons. This protargel is what was used by Bodian as his main research tool. This is the Golgi preparation, a silver stain. The thing about silver stains is that, so first of all, let's look at the silver stain here. Pretty amazing picture. This is a single neuron, all right? Here's the cell body. Here's the axon. Here's the dendritic tree. So let's just marvel at this thing in a couple of ways. Number one, at the massive size of the dendritic tree, and the fact that all of this information is going out that. Okay, so that's the first thing. The second thing is that what makes the Golgi prep successful is how few neurons are stained by it. If you had 5 neurons stained, you couldn't see a darn thing. You'd be looking at black. Okay, you couldn't see anything. Okay, so it is important and intrinsic to the technique that only rarely is a neuron stained with this silver prep. That's an important part. Look at Bodian's. See how multiple cells. The selectivity of the Golgi prep is not very good for connectivity, but mighty good for neuronal architecture. Each cell, each staining, each technique has a purpose. Okay, these are just some other silver stains that I like. This is, I believe, Ramona Cajal. And these are others here. Here's an axonal terminal. And so you can sort of see the cell. Here's the nucleolus and nucleus. And you can see synaptic terminals in this particular silver chain. Look how nice. And you can also see that there are synaptic terminals on the cell body as well as on the dendrite. So, all right. This is from Bob Moore's collection. My neuroanatomy mentor was, I had several. Len Hamer was a great neuroanatomist, and then Bob Moore, my first chair. This is from Bob Moore's collection. Bob Moore was first, he described the suprachiasmatic nucleus and also one of the catecholamine neuroanatomists early in the 60s. And here we have tyrosine hydroxylase. So this is a histochemical stain for an enzyme, all right? This is then dopamine. This is a fluorescent stain for dopamine per se. And here we are in the reticula, in the substantia nigra, pars compacta. And here we are up in the caudate. And that you can see that here we have cell bodies and a few axon terminals, and here we have no cell bodies and just myriad axon terminals. This is transmission electron microscopy, all right? We're going to come back to this neuron and look at the pieces in a minute, but I just wanted you to see the sort of what a transmission EM of a neuron looks like. Here's the nucleus, here's the neuronal, the nuclear membrane. You can see mitochondria, you can see fibrils running, we'll talk about these, rough endoplasmic reticulum, smooth endoplasmic reticulum, Golgi apparatus, all of that coming in here, ribosomes, all of that being present in transmission. That's the introduction to histologic techniques, okay? Any questions?
Are there types of electron microscopy?
Say again?
Are there types of electron microscopy? I didn't know there is a
So
transmission.
there's I'm going to show you scanning
Okay.
in a couple minutes,
Scanning.
but for the most part, those are the two types that I would say. You can do not radiolabeling, you can do histochemistry on electron microscopy for certain. You can do track tracing with electron microscopy and see where the HRP ends up. But fundamentally, there's transmission and there's scanning. All right, let's talk about neurons. Now we're talking about neuronal shapes and types. That'll occupy us for a little bit here, a variety of neurons. I'm going to start with the bipolar neuron, which has axons going in both directions away from the cell. These are largely sensory neurons. You're rarely going to encounter this in any other situation. There's a couple of exceptions that I'm going to show you in a minute. Then you have a unipolar neuron. This is what's known as a unipolar neuron. It has a single axon leading away from the cell body, but very often then that single axon splits. And so it's often referred to as a pseudo-bipolar neuron. And then you have the neurons that you're most familiar with your multipolar. We're going to look at them in a minute. You will see the terminology Golgi type 1 and Golgi type 2. This is very simple. Golgi type 1 neurons are the neurons that have long axons that project to other parts of the central nervous system. The classic example is layer 3 and layer 5 pyramidal neurons of the cortex. And by the end of this course, that will really mean something to you when we talk. But there are others. You can have Golgi type 1 neurons coming out of even the subthalamic nucleus heading over to thalamus. We'll talk about all those. A Golgi type 2 neuron ramifies locally and sometimes very extensively. So it may give off a single axon, but that axon may ramify broadly, and it may contact hundreds or more neurons right locally. Those are going to be your interneurons, okay? So projection neurons and interneurons. Golgi type 1 is basically synonymous with a projection neuron, Golgi type 2 with the interneuron group. I'm going to show you a series of pictures here on different types now, just expanding on what we've said, that we can just look at what they are, all right? Pictures. Here's a olfactory neuron. Here's the dendritic zone, which is the olfactory, in the olfactory bulb. These are the receptors for the actual chemicals, all right? And then they transmit. This, of course, is central nervous system, not peripheral nervous system. And they transmit it down to the synaptic terminals. There are synaptic terminals in the trigone of the brain or other region. Here's the auditory. These are the hair cells of the ciliated hair cells of the auditory. We're going to look at these in extreme detail. We'll have separate lectures on all of this. Here's a cutaneous receptor. Its action potential will come out of there at that first node of Ranvier. all right? This is a pseudo bipolar, right? Single axon splitting. That's what we're talking about. Axon heading out to here, axon heading to here. This is now we get into the world of the traditional multipolar neurons with a big dendritic tree, axon terminals.
Is there a reason it's cut off there? Is there a reason?
No, it's just indicating the concept here is it's much longer than
Okay.
we can show you on this picture.
Okay. That's
all that it means. All right, let's look at lots of neurons in the central nervous system. Here we have a nodose of neuron from the stellate ganglion, right? Bipolar. All right. Here we have a pseudo unipolar. So this is really bipolar, proximal and central. All right. That is, it's not a unipolar. Now, here we have a unipolar. This is a unipolar dorsal root ganglion neuron. One going to the periphery, one going centrally, as approximately. One going to the periphery, one going centrally. And then here's the unipolar trigeminal ganglion, unipolar, one going per periphery, one going centrally. If you remember in our axial localization talk, remember at the very end, we said we played a game and talked about the trigeminal ganglion being analogous to the dorsal root ganglion. Remember how we said that? Form follows function. We're dealing with neuroanatomy. It is no accident. Look how similar these two neurons are. Okay, right? You see, this really is an analogy that works neuroanatomically. Here's a neuron from the intermediolateral column of the spinal cord. Here are a couple of neurons from the superior cervical ganglion and a couple from the stellate ganglion. Notice now that they're starting to show you the axon and the complexity of the dendritic tree. I'm going to just keep going. The sequence is what makes the point. Here we have a neuron in the inferior olivary nucleus. And when you look at this neuron and the dendritic tree in particular of this, okay, this is very stereotyped, isn't it? This isn't just a random pattern. There's a reason. We'll have to explore what that reason is, but these little curlicue hooks, they're there. They're telling you something else is going on. Their form of interaction with other cells is going to be very specific to have such a stereotype pattern. Here is a lovely neuron, the dendritic, the granule cell of the cerebellar cortex. We're going to study cerebellar cortex, which is probably the most amazing piece of the neuroanatomy that we will study is in the cerebellar cortex. And this is a major contributor. This is basically like the phone wire. These axonal arbors are going to travel along the Purkinje cells and contact hundreds of thousands of Purkinje cells. It's going to be amazing. Here's a small cell of the reticular formation of the brainstem. Here's a large cell of the reticular formation of the brainstem. Here's a small cell in the substantia gelatinosa of Rexhead area 2. Here's an ovoid cell from the nucleus solitarius. This cell, when we talk form follows function, this is an amazing cell. I want you to stay with this for a minute and really focus on this cell, single cell. Look at the dendritic arborization of this thing. This cell explains why pain is so poorly localized in the spinal cord, okay? Because this thing is about 5 segments long. And so its input is coming from about 3 to 5 layers, segments of pain information. So right from the start, this is not going to be localized. Pain is not going to have the kind of localization that 2-point discrimination is going to show you. It's just the neuroanatomy reflects. I don't want to say it drives I actually think the brain is more of a physiologist. I think the neuroanatomy has evolved for a purpose. But when you do, form follows function. Here's a large cell from the trigeminal nucleus, and this is a lovely neuron from the putamen. Now, to me, look how fuzzy that looks. Why is that so fuzzy? Why doesn't that look like that or like that? Why are these dendrites so thick and fuzzy? Because they're covered with spines. This is a spiny neuron of the putamen. This is a type of neuron which is involved in Huntington's disease, right? Each of these represents a spine. You can't see it, but I want you to begin to imagine the complexity of the decision that this cell will be making, summating, temporally summating, spatially summating, in order to send out an all or none pulse through this little bitty axon. Okay, this is how the nervous system works. Look at this. This is from Ammon's horn, CA1 of the hippocampus, and the cell body sits between two layers. All right, two different pathways that are coming in. It looks like someone from Toulouse-Lautrec, the woman from Toulouse-Lautrec with a fan on her head and a low skirt, skirt, and here she is receiving information from both. Okay, both incoming pathways, entorhinal pathway and Schaefer-Palato. Here's a thalamic neuron. Look at this guy. Think about the number of neurons which are going to be able to contact this cell, this single cell, which is then going to be responsible for integrate. And not all of these synapses are going to be equivalent by any means. Okay, in fact, they're all going to be different. And here's a neuron from globus pallidus. And then watch this. Here's your Purkinje cell, and here's the pyramidal cell of layer 5. These are just stunning neurons, and this telephone, this system, is then going to head out there. All or none. Okay, all this cell can do is fire a impulse of a single magnitude. Hey, out it goes. At a variable frequency with variable pauses in between in a variable pattern. So it's like a Morse code key. That's all you get for all of this incoming information. It's amazing. Now I put this picture in for a couple of reasons. We're trying to enter an age of artificial neurons. And yet, if you look at the complexity of this, let's just think about this. And here's our first, we're going now into the parts of the neuron, and we're going to start in the dendritic tree. All right. This is scanning electron microscopy. This is transmission electron microscopy. Obvious difference. They don't look anything like one another. You have higher resolution. You can achieve higher resolution on transmission. This slice is about 65 microns in thickness. Okay. It's Bob Moore used to say that if you took all tissue which has been analyzed on electron microscopy, that has ever been analyzed from beginning to the end of time, all of the tissue put together could fit in a sugar cube. Okay, so you have a sampling problem when you're dealing with electron microscopy. That's intrinsic, and yet you can get up to 250,000 magnification. All right, so you can get almost to a molecular level with transmission EM. All right, scanning EM, on the other hand, shows you structure, shows you a beautiful organization of structure. Now, also for a very, very small region. Now, let's just look at this for a minute. So here's what we've done. All right, let's just talk about this because we're going to use these photos over and over. We took the brain that I dropped into liquid nitrogen. And then I took two plates, okay, and I put those plates and I picked up, they're also at liquid nitrogen temperature, and then I squeezed the brain against these liquid nitrogen cooled plates. And then with a device that could really generate a lot of force, I snapped them apart. And I now have a face, an A face and a B face, okay? Now I can take that face. I'm not going into it. I don't care what thickness it is, okay? I'm only looking at the surface of this thing. I could have taken a piece of tissue if I wanted. I didn't have to do whole brain. I could cut out a piece of tissue and do the same thing with it. But when I'm done, I now have this surface, this irregular surface where the huge force of this machine separated, pulled off. If there were any spines, here's we caught a spine just in the right plane. So we can see that it's leaving the dendrite, that it has a neck, that it has a bulb at the end, okay? Here, we just ripped the thing off, okay? All of these then are just ripped fractions. Now, and here's a membrane. We left part of the membrane on the dendrite and the dendrite lost part of the plasma membrane. So here we're looking at the membrane. Now the next question is, how did you create contrast? Because okay, we crack it, well, we've got this liquid nitrogen tissues. Like, well, so now what you do is you take a gun, which is just going to spray atomic gold at a very low angle and create shadows. Now, normally, so if we were shooting light, then the bright area would be where the light hits, and the dark area is where there is no light. No, we're doing the opposite. We're creating dark by firing the gold atoms at the tissue. And so we're actually coming from this direction, from bottom up, not from top down on this neuron. And the shadow is this part, which didn't reach where all the gold atoms were prevented from reaching. Okay, so here you caught a bunch of gold atoms. All right, good. So that's what we're going to be looking at. These are dendrites, and we'll come back to, well, we're in the dendritic area. You can see these equally spaced in the dendrite. You can see the neurofilaments equally spaced from one another. You can see mitochondria. All right, they're pretty large. You can see that on the dendrites, there are synapses. We're going to look at this at still higher magnification by a lot. Here, just to anticipate, we've got lots and lots and lots of synapses, synaptic terminals, boutons, meeting on much smaller pieces of the dendritic tree. Here's a little bit of myelin, right? So here we have a myelinated axon traveling. All right. Right, onto the cell body. The cell body of a nucleus of a neuron is fairly large. Our ruler is the 7-micron erythrocyte. So it could be smaller than a red cell, or typically 2 to 3 times. This is the cell body, 2 to 3 times the size of the red cell. The nucleus, this is the nucleus. So the nucleus is actually larger than the red cell itself. The nucleus of a neuron on most light level techniques is pale. It's well centered in most cases within the cell body. The oligos, for instance, we'll get to those. They're like fried eggs, right? We'll talk about that. But it's centered in the center of the cell body, the most neurons. You then have in the center of that a nucleolus, which is going to be filled. It's a non-membrane bound structure containing tons of DNA, RNA, and nuclear proteins. And then, sometimes I'll just point out these peculiar things. You have this very weird puckering of the Purkinje cell nucleus where the RER is. Again, we'll look at it. Here's back to that picture, and you can see the relatively clear nucleus with a very dense nucleolus in the crescent-violet stain. Other studies might not do that. And rough ER all over the place, filling the cytoplasm, but not the nucleus. Here's that Purkinje cell nucleus. We can see the non-membrane bound nucleolus, collection again of RNA, DNA, and nuclear proteins. There's a little collection of probably transport proteins. When this picture was taken, they had no idea what it was. Here's the Nissl body. These EM, most of the EM are from the EM atlas by Peters, Chan, and Pele. Okay, actually it's Peters, Chan, and Pilati. I went to school with Pilati's son. That's when I was first introduced to the EM. And so here's that peculiar area in the Purkinje cell. We look at the cytoplasm. There's a lot of going on at the EM level in the cytoplasm, or depending on the particular counter stain, histologic stain that you might use. You have the plasma membrane, of course, delineates the neuron. Back to the Bodian hypothesis, it's an individual cell, not a network. You have this Nissl substance we've been talking about. You have then the Golgi apparatus, which is smooth ER, specifically for the purpose of glycosylating glycoproteins. Mitochondria are everywhere, everywhere. You're going to see them everywhere. Everywhere that you need to use energy, you need a local supply of energy, and you therefore are going to have mitochondria. The more energy consumption, the more mitochondria. The more mitochondria, the higher the energy consumption is going to be in that area. You then have neurotubules and neurofilaments, and then lots of other lipids and pigments. And so here we have a neuron, and we want to point out, looking at the cytoplasm, the first thing I want to point out is that you clear out this area of the axon hillock. We're going to look at this in a minute. This is an artifact. This is a shrinkage artifact. The cell body shrunk and the neuropill, this is referred to as the neuropill, all the dendrites and axon terminals did not shrink as much. And so the neuron cell body was pulled away. And these are all artifacts. Okay, all artifactual. Huh. Here you have dendrites. All right. Electron microscopy. We're gonna look at a even bigger one, but this cartoon shows you the area that we can look at as nucleus with its limiting membrane, nucleolus without a membrane, RER, SER, Golgi apparatus, mitochondria, and tubules, microtubules, and going out. And this I thought was interesting as I was looking at this picture yesterday. I just want to focus on this. We can't quite see it. We could blow this picture up more, but not here. But look at this. This is a dendrite, right? This is a dendrite, not an axon. This is a dendrite. And this dendrite is then bifurcating. And look at this machinery complex that exists at the bifurcation. Because you know what? This cell is going to have to send proteins and assembled, not just proteins, but assembled membrane-bound particles out, one dendrite or the other. And they may not be wanting to send the same material everywhere, okay? They may not, these dendrites may be contacting different neurons, receiving inhibitory or excitatory information. So they need to have, and so there is this, I never noticed this before, but there's this whole assemblage that is sitting here at the point of switching, if you will, of transport switching. Very interesting. Or receiving, you could put it the other way, or information receiving. Here's a little bit bigger. When we look at it up this mag, this is probably not 100,000 yet, but in the range of 100,000. So again, nucleus, chromatin, right? Chromatin, mitochondria, rough ER, smooth ER, right? Golgi. This is what Golgi looks like on mitochondria, lysosome, dark body, probably lysosome, mitochondria, rough ER, ribosomes. Not a lot of glycogen. We'll come back to that issue. I do want to return to that issue. In fact, we're going to end with that issue. And coming out here, you get a sense that there are now something that are some structures that are running out here. So fibrils of some sort. We're going to come back to those. The axon hillock is the usual site of impulse generation, and there's no RER in the axon hillock. It seems as if when you really want to get specialized about what you're going to do, so there are certain things that can be present and certain things that can't be present. And you don't have RER in the site of impulse generation. Otherwise, you have pretty much everything else. The thing is surrounded by a plasma membrane. We keep saying that. Don't forget the plasma membrane. It can be the axon hillock, can be myelinated or not myelinated. It can go either way. It's usually a very short distance from the cell body. It is in the periphery, it's going to be the first node of Ranvier. The nodes of Ranvier are created then by the myelin, which cover most of the axon, but not all of it. This is an important topic of how the oligo knows where to insulate and where not to insulate. So we'll have to take that up. We're not going to be doing a lot of the chemistry this year, but we're going to lay the groundwork for the anatomy of it. Right? We will have a separate lecture on glia, including myelin. And then I want you to think about the process of myelination. In the periphery, you have a single Schwann cell investing a single axon. In the central nervous system, the oligo may be contributing to myelin of 10 or more neurons. So the idea of the cell body of the oligo wrapping around is going to get very messy if you try that. That's probably not going to work. Go to the pictures. Here we have a cell body, right? Pericardia, nucleus, nucleolus. Nucleus is centered to nucleolus, and in this case is not, okay? No RER. This is the axon hillock. So very nice picture. No rough endoplasmic reticulum. There is a plasma membrane binding it. Okay, good. Here's a dendrite. This particular dendrite also has no missile substance. I'll use that term on purpose. And there's a missile substance. Here's a EM of a localized dendrite. There are neurotubules and there are neurofilaments. We're in the axon here that participate in the exoplasmic transport. We had a very prominent set of discussions in our neurochemistry section last year on neurofilaments and neurotubules. We're not going to do that, but I'll show you pictures. I want you to see them so that you'll recognize them when we look at EM. All right, and they're going to transport. They're responsible for antegrade and retrograde axonal transport. The axons then terminate in the synapses, which we'll look at. I've chosen a silver stain for this one for you to see axon. This is the axon, I believe, and here it's myelinated, and here it's not myelinated, and here it's not myelinated, and this is the axon hillock, and these are dendrites, and that's just a nice picture, not a Golgi prep. Here's a series of different preps just designed to show you the separation of axon from myelin. In this case, the myelin is simply nothing. The lipids have been removed. And so we just have an axon being stained with nothing surrounding it. Okay. I don't actually know that the lipid is gone. I only know that if it's here, it's not being stained. So we've used a selective stain for the axon and to avoid the myelin. On here, we've sort of done the opposite. Okay, here we wanted to see the myelin, okay, without staining the axon. And so you can get a sense of thickness, and you can get a sense of thickness. And these are longitudinal. Most likely, this is from the periphery, if I were to guess. And this one of the pictures from last year, just showing you the idea that there are neurofilaments and there are neurotubules. The neurotubules are tubulin-based. They are classic microtubules, 25 micron wide structures that are made up of the tubulin monomers and the myelin and the microtubule associated proteins. They're MAPs. Your MAP proteins are all on the microtubule, okay, including tau proteins and things like that. So we want, while we don't talk about clinical, this course is designed to give you the basic knowledge that you can interpret the pathophysiologic information that we're given by researchers. Neurofilaments in the central nervous system can be cross-linked, heavily cross-linked, as in this picture, or uncross-linked, as in this picture. And so this is a transmission, right? And these are both scanning. So the scanning can be done in an incredibly high mag also, all right? And gives a very different kind of data. It would be hard to come away. It would be hard, for instance, to see the difference between these two on a transmission EM. You might get a sense of some decoration of things leading away at right angles, but the odds of their traveling in the plane of this 65 micron section long enough for you to see it is, like even if they're there, I mean, look, they don't travel straight out. They go at an angle, in which case you're going to leave the plane and you won't see it. So a huge advantage to the scanning EM. This is very difficult to do. It's a tremendous tour de force. Transmission EM can be spectacular. This is absolutely one of my favorite pictures of all time. I use this picture over and over again. This is a node of Ranvier from the atlas that I'm talking about. Here's the node, devoid of myelin. Here's the compact myelin. This is also part of the myelin. These are the filopodia. Each of the layers has that cytoplasm containing bulb at the end, and that's these. And look at the structure of the interacting proteins with the axon, the interacting structures. And each of these, of course, is a layer of myelin. And this is the cytoplasm of the myelin, which is extruded in the compact myelin, but visible here in the terminal. And then you have beautiful pictures of microtubules and neurofilaments. Neurofilament is 10 to 12 microns, and neurotubule is 25 microns, classic microtubule. Okay. And again, back to those pictures that we saw. Now we're just showing you synapses. And this is synapses at the light level. And these were called boutons, all right, from the French term bouton. And we still call them boutons. I still call them boutons. Not everyone does, but they're terminals, axon, terminals, synaptic terminals, synaptic buttons, synaptic, whatever you want to call them, synapses, simply call them synapses. All right. This is a cartoon of the synapses, and you would like well, I'll turn it around. We learned this cartoon structure from electron microscopy. And so the cartoon is showing you what we've learned. That is that there is this active pre and postsynaptic terminal. This is a synaptic cleft, which is present, all learned from EM, all right? That you have synaptic vesicles, which cycle detail. We did this in huge detail last year. That there are, of course, mitochondria. This is a energy intensive. That you don't find microtubules or neurofilaments in the synaptic terminal. Partly we think because they're busy doing other things, partly because they're plastic. They're constantly changing, and the microtubules and neurofilaments tend to inhibit plasticity. Neurofilaments in particular are very slow to assemble and take apart. Microtubules, instantaneous. So in embryology, when the neuron is finding its way with its pseudopods and looking for traveling down the chemical gradients that it's using, okay, identifying receptors as well as small molecules and migrating to where it's supposed to go, you sure don't want any neurofilaments in there. Microtubules, okay, that can give it some strength. And if it doesn't like it, instantaneously, we'll show you pictures of microtubules disassembling, and it's almost instantaneous. And the philopod can just retract and go, I didn't like that. I'm not going that way, okay, and head off in a different direction. It wouldn't be able to do that if it had neurofilaments in it. Form follows function, okay? What is present and what isn't present is all about what it's doing at that moment. And it tells you what it's doing if you can read the language of the neuroanatomy. And that's a point that I want to make. Why don't you put any clinical in this course? Why am I strict about excluding clinical? To me, because we're neurologists and we're really interested in clinical medicine, and clinical information screams at a high volume. But the language of neuroanatomy and of all of the basic neurosciences is subtle and quiet and very subtle. And if at the same time the clinical is screaming, you don't learn the subtle language of the basic sciences. And that's what this course is about. You're learning a language, but with the language are the concepts that are embedded within the disciplines. Neuroanatomy has its own logic, okay? Neuroanatomy, which gave you this cartoon, I still say that form follows function, but that means that once you can read the form, you can see the function. But if you're only worried about why a person has a Babinski, you're not going to learn this language. And that's why we separate them, okay? Eventually you have to put them together. But first you have to learn the language and the concepts. It's more of the concepts than the language. Here we are in a synapse, all right? Mitochondria, of course, okay? Synaptic vesicles all over the darn place. We'll come back, we'll have an anatomic lecture on the synapse, at least one, all right? Here's a synaptic cleft, there's the presynaptic proteins. We're looking at proteins on the transmission EM here. Here are the postsynaptic proteins. We're gonna get better views of these on when we get to other pictures selectively for the purpose of showing you. No, not everywhere. You don't have that everywhere. There's a contact site, however. We'll come back to that. Look at this view. Look at this scanning out. First of all, this shows you the sense, this one on the left, shows you the sense of the crack that I'm talking about, okay? This one really shows you it. Here we have, let's just focus on what we have here. Here we have a synaptic terminal, okay? We have an artifact. The plasma membrane has pulled away from the cytoplasm. So here we're looking at cytoplasm intact, the structure of cytoplasm. It's not just loose amorphous water. It has a high structure of its own that can be captured on EM. But the plasma membrane pulled loose at the time it was done. Okay, and so there is a separation between them. Here's a mitochondria. Okay, here are synaptic vesicles. If we look closely, we can see that there are interconnecting proteins or interconnecting structures between the synaptic vesicles. It's like, holy moly, here's a synaptic cleft, and we can see, not only can we see the density of proteins in the cleft, but oh my God, here we are in a dendritic terminal, right? The bulb from the dendritic spine, and we're looking at the receptors which have organized themselves into the synaptic terminal, okay, into the synaptic cleft. These receptors are not spread out throughout the terminal, okay? They are brought together to create an assemblage, in this case, a two-dimensional assemblage in the plane of the synaptic terminal cleft. This is the kind of things that we're going to be dealing with in neuroanatomy. And here you have the transmission, which shows you the same thing, right? Now you can see the relationship. And the osmium tetroxide quite heavily stained here, plasma membrane, you can almost make out the bilayer. Here's some weird membrane system, probably an artifact, but could be a multivesicular body assembling. A probable mitochondria, synaptic vesicles, all eager to contribute their neurotransmitter small molecule into the synaptic cleft upon receipt of the calcium signal and right? Yeah, you gotta love it. Now, here's a dendrite, and of course the obvious thing is the mitochondria. And this thing is receiving synapses from all over the place. So we're just seeing a few of them here. There are more and more, but here's a point. Keep looking at this. Here's your mitochondria. Very busy, right? So it must be very busy. It must have an enormous metabolic demand. Here's your glycogen store, here and here and here. Am I my brother's keeper? Who's maintaining this? Where does the mitochondria get its energy supply from? So I get it, they're making the ATP for us. Okay, but where's the glucose or lactate or pyruvate coming from? There's no, this dendrite is filled with mitochondria, okay? And we don't see any kind of apparatus here to generate the store, the substrate that is needed here. So that's the question, is how is there enough glucose? How is there enough oxygen for this cell to run? Okay, and my answer is not from the neuron. Okay, the neuron is so specialized that it can't maintain itself. Okay, therefore, the next lecture is about glial cells, and we're done. All right, fine. For those of you who may be seeing this outside the room, this photo is taken from a collection of photos by John Lund, and I should hasten to add that all of the photos, none of these are directly mine that I can think of in this particular lecture. And so they've all been taken from others. I do that simply on the basis of that this is only for education, and I will never sell these lectures, which are open source for everybody to use. And John Lund, who I've never met, gets credit. You can look him up on the internet. And thank you. We've finished lecture 1. All right, next week, the glial cell. Or glial cells.